Most of us have seen pictures like this. We remember this from medical school, the karyotype. And so for me, this is like the first digital camera. Remember the first digital camera? It was exciting, but it was so grainy. The resolution was really poor. So this is like, the first digital camera. Wow, look, and unless there were massive abnormalities, like a trisomy or a massive deletion, you'd get karyotyping done and go, oh yeah, it looks normal. The 0.1 megapixel image is fine. The X-ray of the brain, it looks normal. So it didn't really translate, but that's not surprising. It only translated for gross abnormalities, mainly seen in syndromes presenting to paediatricians. What high resolution means is single nucleotide polymorphisms. So, and I encourage you to try and jot some of these things down and remember this. All you need to know is just a handful of things to be competent in this space, I'd say, as a clinician. So this is the most common type of genetic variant between people. It's a single variant in a base pair. So you'll know the nucleic acids. It's just one change. And it happens in about 1 in every 1,000 base pairs. So we needed to get to a resolution where we could see the smallest little bit of DNA change to say, okay, we've got some resolution happening. And there are two types, synonymous and non-synonymous. So a synonymous one means it doesn't seem to change the gene expression, the outcome, the phenotype. A non-synonymous one, it seems that little variant leads to different outcomes. And it's obviously the ones that might lead to different outcomes that might have clinical utility. So SNP, single nucleotide polymorphism, synonymous, non-synonymous. So if you're looking at a paper that says, we found this SNP that does this, but it's a synonymous SNP and we don't know what the hell is going on, maybe put that paper lower down the pile or consult a colleague. If they say we found this SNP and it's associated with this, and interestingly this SNP really changes the gene expression and tissue, and so it's plausible, maybe put that higher up the reading list. So I'm trying to give you some basic metrics on how to make your own decisions about the literature. Because I think as medical practitioners, that's part of our core role, is to think with our own minds. So if you've got something which has got a synonymous SNP as their finding, it's like, well, maybe it's a spurious finding, a statistical fluke. Or maybe we haven't figured out exactly how it works to alter gene expression. There are other less common genetic variants, copy number variants, deletions, insertions. These are less frequent but tend to have a much bigger impact. So when you look at, for example, literature on autism spectrum disorders, when you look at conditions that seem to have a higher prevalence correlating to age of paternity and maternity, it's probably more of these microscopic abnormalities where little chunks of DNA are blasted out by cosmic particles. The cosmos is playing with our DNA. It's a fact and it's pretty cool as well. And then people say, why? And well, it's called evolutionary design, it's trial and error, some random errors. Occasionally that equals a longer giraffe neck. Sometimes that's useful. So often people say, oh, why have I got this unhappy variant? I'll say, well, blame Mother Nature. It's kind of how we're designed. So you'll see studies where they say, look, this is a very rare gene variant. It's a deletion or copy number variant, but it has a massive impact. 80% of people with it get schizophrenia, or 99% of people with it get a problematic autism spectrum disorder, but only 0.1% of the population has it. So this is another key sort of metric to analyse papers. How common is this gene variant? Does it affect every other person, or 1 in a billion? And what's the impact clinically? And so this is something worth thinking when you critique for yourselves literature. Variable number tandem repeat. So things like Fragile X syndrome and certain things. So sometimes there can be extra chunks put in through a duplication error. So that's different from the sort of cosmic particle knocking off a bit. But there can be duplication errors where bits are not included. And it gets complicated, but big chunks that vary. It happens rare, but it can have a big impact on outcomes. Now, haplotype, all a haplotype is a section of the DNA strand that tends to travel together. It's inherited as one block, right? That's all it is. That's all it is. So if there's a whole string of base pairs that tend to travel together through the generations, you can just analyse that as one chunk of heredity, and that helps with your statistical power when you're doing studies. So that's all a haplotype is. And haplotypes are these chunks that travel together, and they might have different SNP variants. So often in a chunk that travels together, they'll find a thing called a tag SNP. So a SNP that goes along with that chunk that they can use to measure haplotype A or haplotype B. So that's all a haplotype is. So when you're reading a paper, they say this haplotype was different to that haplotype, blah, blah, blah. It just means a chunk that travels together intergenerationally. You'll remember this. So if only it was as simple as a simple sort of helix like that, we could read it like a needle on a record, you know. I always imagine the DNA is just this strand that you could read, you know, A, C, T, G. That's what it really looks like. We're nowhere near figuring it all out. Nowhere near. It's exciting, but we're nowhere near. So in reality, there's a very complicated wrapping up process, and the wrapping and unwrapping process can vary between individuals and be passed intergenerationally. So it's another mechanism of heredity. So this is all the epigenetics. A big part of the epigenetics is around this, the wrapping up of the DNA, because unwrapping it's necessary for the ribosomes to then produce a protein. So wrapping, rewrapping is another whole layer of complexity which we're really in the infancy of understanding. And this slide sort of shows that you've got the chromosome, then there are supercoils within the chromosome, then there's further coiling within the supercoil, then there's one coil within a supercoil, you know, then there's the DNA nucleosomes, and then if you unravel that, then you get that simple thing. So it's a lot more complicated than just the sequence, and figuring out this, there'll be multiple Nobel Prizes to come from this, and this is what epigenetics is really looking at, things that affect the coiling and expression environmentally of DNA. And so a lot of you would have probably heard of people talking about epigenetics. So it's really how coiling issues and also methylation, so chemical modification of sequences through environmental factors can alter gene expression. That's all it is. That's all it is. So histones are what the DNA is coiled around when it's bundled, and they can have modifications that affect how tightly they hug that DNA or release it for transcription. And there are various chemical processes, but it's a very early science compared to the straight SNP sequencing stuff. What you need to know specifically is methylation. So methylation is certain genes can be switched on or off based on methylation of a thing called CPG islands. So basically this is the switching on and off of genes, like serotonin transporter link promoter region, that can be switched on and off depending on environmental stress. So that's all it is. So if somebody says, oh look, there's this SNP association, you can always say, well look, was it stratified by methylation? And of course, this wasn't done in most studies. So there'll be another whole wave. It'll be a slower wave. A single methylation study is about 500 US now. You can get your whole genome for about 800 US now, just the base pair sequence. So there'll be a longer gestation before the price point drops. There are then heaps of research studies that can be done to show clinical validity, utility. But that'll be the next wave. And there may actually be more signatures coming through. Based on looking at methylation, is it switched on or off, then we analyse the results. That's all you need to know, methylation, epigenetics, coiling. So the CASPI study, and you should know CASPI, so basically they were looking, New Zealand-led study, looking at stressful life events, odds of depressive symptoms later in life, a big cohort, looking at gene variants, and they found that the short version of the serotonin-related gene variant, the serotonin link promoter region, seemed to be associated with higher odds of having depressive symptoms in function of number of life events. So it's almost like the situational stressors, childhood traumas, and that genetic susceptibility. And they seem to have a dose effect depending on if you had the long or the short version of it. So my read on it is that the long version is longer, so the ribosomal transcription mechanism has got a longer area to land on to transcribe transporters that help you recycle serotonin. That's just my making sense of it. Nobody can directly prove that, but it sort of makes sense. They also got data looking at suicide. So if you had this short version of that serotonin transporter bit, and number of life stressors, probability of suicide ideation and attempt went up. Of course, they couldn't use actual suicides because happily they're relatively rare, so you'd never get a powered study. But I'll come back to those slides later. But so that ends. This slide ends kind of the backgrounding. So hopefully, you've got a sense of DNA, base pairs, SNPs, methylation, gene on, gene off, epigenetics, environment, coiling. And just read it again and again. Read it 10 times till you no longer get the slight palpitation. Then you're ready. That's all I basically know. I don't know that much more than that. I mean, I know some more, I think, but that's the guts of it. So this is not low level stuff. If you know this and get over any anxiety around it, you're kind of weaponised to then read papers and advise people.